Start in just a moment. Hello, everyone, and welcome to the second webinar of the APDA Connecticut Chapter Educational Webinar Series. My name is Marlene Argianis. I'm the Chapter Coordinator for the American Parkinson Disease Association Connecticut Chapter. Thank you for joining us today. Before we get started, I just wanted to say that our prayers and thoughts are going out to all those people in Florida and the family members. We hope that they all stay safe during this difficult storm. We are extremely grateful to Dr. Jesse Cedarbaum for volunteering his time to present today's topic, Preventing Parkinson's Disease, the long and winding road that leads to your door. After some brief announcements, Dr. Cedarbaum will give his presentation, and then we'll be answering your questions in the Q&A session. American Parkinson Disease Association Connecticut Chapter, or APDA-CT for short, is part of the largest grassroots network dedicated to fighting Parkinson's disease and works tirelessly to assist more than 1 million people with Parkinson's disease in the United States live life to its fullest. In the state of Connecticut, there are approximately 10,000 people with Parkinson's. The APDA Connecticut Chapter offers a variety of services in the state of Connecticut. Please visit our website to explore all that we have to offer. Our website also hosts a vast array of publications, webinars, articles, and information that can help you understand Parkinson's disease. If you are participating from another state, we have chapters and locations throughout the United States. And you can find that contact information on our website as well. The APDA Connecticut is available throughout our helpline, email, as well as our Viva social media outlets. A special thank you to our educational webinar series sponsors. Our silver sponsor is Savonian, and bronze level sponsor is AVI. Please visit their websites for more information about their products and services. A question and answer session will take place immediately following Dr. Cedarbaum's presentation. You can type in a question at any time during the presentation, and we will answer as many questions as time allows. And now to begin our presentation, we are delighted to welcome Cynthia Streeter, a representative from our silver sponsor, Savonian, who will introduce our guest speaker, Dr. Jesse Cedarbaum.
Thank you, Marlene. Thank you so much. And I apologize that I'm not on camera. I'm using an iPad. But I am from Synovia, and as Marlene had shared, and we're so happy to support the APDA of Connecticut. Dr. Cedarbaum is a 1978 YSM graduate. Dr. Cedarbaum is a neurologist, clinical trialist with a subspecialty in movement disorders. After 7 years heading the Parkinson's and movement disorder programs at New York Hospital Cornell, he spent the majority of his career as an executive in the pharmaceutical industry, leading teams in the areas of neurodegenerative, neuropsychiatric, and retinal disorders at the Regeneron, Elan, Cytokinetics, and Bristol Myers Squibb. Most recently, he led a team at Biogen that advanced 3 new compounds into the clinic to the potential treatment of Parkinson's disease. At Biogen, he also established a clinical research program in inherited ataxias. His research activities have encompassed efforts to develop novel clinical outcome measures and biomarkers, including digital health technologies for clinical trials. We are so excited to have Dr. Cedarbaum here with us today. Dr. Cedarbaum, welcome, and thanks so much for joining us.
Thank you very much for your welcome here. I'm very excited to be here. Back in the days when I was at Cornell, my mentor there, Dr. Fletcher McDowell, was the first medical advisor for APDA. So I'm very familiar with the organization, and I have known APDA for a very, very long time. So what I'm going to talk to you about today is, you see my title on the screen, and the reason I'm saying that this leads to your door is because we who develop drugs and novel therapeutics cannot do this without you and your participation. And this is why it leads to your door. So James Parkinson, who lent his name to Parkinson's disease, was a very, very prescient fellow. And he described many of the clinical features we know about Parkinson's disease, and subsequent researchers described the loss of the dopamine neurons in the substantia nigra that you all read about, and the presence of the Levy bodies, the inclusions inside neurons that seem to mark dying neurons, and the fact that these are made largely of a protein named synuclein, and that synuclein, these changes seem to spread around the brain, starting at the bottom and working their way up to the top, which is why people with Parkinson's disease seem to, at least in the beginning, develop new symptoms in a fairly stereotyped order. But Dr. Parkinson was a bit of a prophet, and this is what he wrote in 1817. Unfortunately, we're still working on it, but I think we're getting closer and closer with your help. So here's what I'm going to talk about today. I'm going to talk a little bit about clinical trials and clinical research. What's the difference? About how drug companies approach the drug discovery and development process, the stages in the process, and probably particularly for your interest, why it takes so darn long. We'll talk about the results of the recent synuclein antibody trials that were recently published together in the same issue of the New England Journal of Medicine. We'll talk about how we're using technology, smartphones and smartwatches, to help us track Parkinson's signs and symptoms. And then finally, we'll wrap up with what we're doing here at Yale, and then why it's important. I believe it's important for every single person with Parkinson's disease to participate in research and how you can do it. So clinical trials and clinical research, what's the difference? Clinical research studies are research studies that collect data from and or about persons with the disease or condition for the purpose of better understanding how the disease affects people and how to treat it. And there are several different kinds of clinical research studies. Some are what we call natural history study, simply observing what happens to people over time. Sometimes we collect biological samples or brain scans or other data about the body and how it changes. And sometimes we actually do research studies to test out new ways of helping us gather information. And I think I'll talk about examples of all of these. Clinical trials are a specialized form of clinical research study in which we're testing the safety, the tolerability, that means the side effects, and the therapeutic effects of potential new treatments. And as we know, all too often, these trials end up with negative results. This is a very, very long process. And it's also a process in which there's a lot of attrition. So pharmaceutical company may start out testing as many as 5,000 to 10,000 new compounds. And many, many, many fall out in testing in animals or in the laboratory. A few out of those 5,000, maybe 5 or 0.1%, will actually make it into early stage testing in humans. And we'll talk a little bit more in a slide coming up about the stages of testing in humans. And then out of all that, we might get one FDA approved drug. And a lot of people will say, well, it's all the FDA's fault. They're too strict and yada, yada, yada. I'm here to tell you that I don't believe that is the case. I think the FDA, in my experience, has been my partner in scientifically developing drugs. And as Dr. Dunn, who is the head of the Neurology division of the FDA, says, problem is we still haven't really discovered the right drugs and powerful enough drugs to do what we want them to do. And we're keeping on working on that the whole time. So once we get to testing of drugs in patients, there are 4 steps. And you may have heard these terms. Phase 1 tests are very small tests, small trials. They'll involve maybe up to 100 people. They'll last up to a year and a half or maybe 2 years. And the goals are really to understand, is the drug safe to give to larger numbers of people? Are there any very bad side effects? Because if a side effect shows up in a small number of people, you bet it's going to be a big problem when we try to give it to everybody who has the disease. And also to help us understand how often to give the drug, what the best form is, et cetera. It's pills, shots, whatever. In phase 2 studies, which will enroll 100, this means, does the drug? In phase 3, these are definitive clinical trials to demonstrate that the drug actually works and that it's safe and it's good to give over the long term. These are the trials that ultimately will result in approval by the FDA or the EMA in Europe or similar bodies in other countries. And they involve over 500 to 3,000 patients, for example. They can take many years because it takes a long time to find these people and because we want to watch what happens to people over periods of a year or 2 to make sure that the drug is really working. Once a drug is approved, companies may do what they call phase 4 studies. And these are studies that may be required by different regulatory bodies like the FDA, or they may be done by the manufacturers of the drug to try to understand how the drug is behaving in real life, in real world situations, to more fine tune who should be getting the drug and how it should be used. So 4 phases of drug development. So how do we go about doing all this, putting all this together? Well, first of all, we need to know what we're using a new drug for. So in the Parkinson's disease world, we have at least 3 classes of drugs that drug companies are working on. First, the one most of you are familiar with are the symptomatic drugs. These are medications that are designed to treat specific symptoms. They usually act quickly and they often wear off quickly, but they don't slow the overall course of the disease. And examples of these drugs that you're familiar with are levodopa or Sinemet, dopamine agonists, Azilect, amantadine, drugs like this that your doctors have probably prescribed to you. Now, in the last 20 years or so, the goals have gotten a little loftier and we talk about disease modification. These are drugs that work over time to slow or halt the progression of the disease by working on the changes, the pathological changes that are causing the disease in the body and prevent worsening of symptoms. These can interfere with genes in the way they work or proteins that are believed to cause the disease. And the examples are some like I'll show you today are the antibodies and other drugs that are currently in development, gene therapies, et cetera. And the last category are still, I think, far off. Unfortunately, these are reparative and restorative therapies whose goal is actually to restore function that has previously been lost. And examples of this might include transplants or cell-based therapies or some of the gene therapies to put back a protein that has been damaged or missing in the course of the disease. How do we do this? Where does all this information come from? So we start with people. This is called bench to bedside, we call it, because we're taking data from people, from our clinical experience, from watching the disease as doctors, to understanding of the biological pathways and the genetics. And then we use this data to inform laboratory experiments, looking at biochemical pathways, at pharmacology, which means how drugs work in the body, at genetics, certain genes we might be able to target. You've all heard of these. We'll talk about a couple of them in a moment, as well as understanding whether the drug is safe and how finding what we call markers, which are changes in the body that show us how well a drug works and help us track it in the body as we study it. And all this takes a long time. So there are at least 9 steps that what we talked about in drug development. Identifying what compound we want, what target we want to go after, finding a set of drugs and compounds that might work on it, finding the best one, and then getting ready all the things we need to do to have a drug that we can put into people. Then the 3 stages, the first 3 stages of drug development I talked about, and finally filing with the regulatory authorities for approval. And what kinds of things are a drug company interested in finding out before they put a drug even into people in the first place? Well, what kind of drug do we want to use? Is it a pill? Is it a shot? Is it cells even? Does the drug have to get into the brain? Does it really work well in what we call binding or getting to the target where we need it to get to? How long does it stay in the body, which tells us how often we have to give it? Is it safe? What are the side effects? How well does it play with other drugs? Does it cause problems? Does it help some other drugs, in fact, work better? And are there tests that we can use to help monitor its use in the clinic? We want to know which patients it's best for. Eventually, we need to know how well is it going to work compared to other drugs. And are there other drugs that do the same thing? Because for a pharmaceutical company, obviously, you either want to know whether you have competitors or whether you need to position your drug to work together with something else. A good example of that, you may have heard recently about a drug named apictopone, which helps Sinemet work better. So that's an example of what drugs working together. And again, this is a long, long process. Just the part of which where we're selecting the right drug and dose can take between 3 and 10 years. And then in the patients, selecting the right patients and showing it works can take another as long as 7 to 10 years. Very, very long period of time. And part of the reason it takes so long is it takes a long time for us to find the patients, to participate in these clinical tests, and then we have to watch and wait and see what happens over periods of 6 months, a year, sometimes longer, as long as 2 years. It just takes a long time to collect all that data. When people ask me if there's one tool that I wish I had for doing drug development, what would it be? I always say a time machine, because that's the one thing that could help us speed up this whole process for all of you. So let me talk about some of the recent synuclein antibody trials, but first, let me just talk about the whole space of drug development for Parkinson's disease. In the Journal of Parkinson's Disease recently, there was this very excellent summary that tallies up all the drugs in development for Parkinson's disease. And on this very important website, and I'll give you the link to it at the end of the talk, called clinicaltrials.gov, where all clinical trials in the entire world are registered. There are 147 clinical trials currently registered, or were when they wrote this article about this time last year, in Parkinson's disease. The vast majority of them, or 62%, are still testing symptomatic drugs, and about 40% are testing drugs focused on disease modification. About 35% are in phase 1, about 50% are in phase 2, but only 15% are in phase 3. And of these, at the time they wrote this, only 3, and we're going to talk about 2 of them today, of these trials were in potentially disease-modifying therapies. 42% of all these clinical trials are testing totally novel drugs. About a third of them are testing approved drugs that have been what we call repurposed, that there's some scientific reason to believe that they might help Parkinson's patients, but they're approved for another disease. So these are being tested in Parkinson's disease. And about 20% are trying to improve on what we call the formulation or the dosage form of existing drugs. And if we break these down by phases, we see again that a big mixture of different types and different targets, everything from the microbiome, the bacteria in your gut, to drugs targeting alpha-synuclein, to cell therapies. All of these are being tested in the very, very earliest phase, 50 of them in all. In phase 2, we have 65 drugs and 64 trials, very, very much the same spectrum, but we're still catching up in phase 3. And I imagine this profile is going to change over the next several years. And Mr. McFarthing and his colleagues, I think they've pledged to put out this paper an update on this is every year, and it's really worth following to see the progress. So what are some of the gaps that we have in treating Parkinson's disease? Let's focus first on symptoms. We have still a long way to go in treating the basic symptoms of Parkinson's disease, the bradykinesia or slowness, the rigidity or the stiffness, the tremor, the on-off, the fluctuations that I'm sure many people, and the dyskinesias that I'm sure many people on this call experience, and then the symptoms of late disease, such as falling and cognitive impairment. A lot of ways to go in all of these, as well as other what we call non-motor symptoms like sleepiness and fatigue. And potential treatments for all of these are currently in the pipeline. And we don't have time to talk about all of these, but this is just a list of a few of these and the companies that are working on them. So this is very, very, very encouraging. And then one of the ones that I find most encouraging, particularly for people with fluctuating responses to levodopa or on-off phenomena, are 2 new drugs, and these are actually reformulation drugs that are approaching approval. One is from the company AbbVie, and the other is from a company called Neuroderm in partnership with Mitsubishi. And with these are our infusion pumps that will pump levodopa under the skin so that you get a very, very continuous profile. So normally when you take a Sinemet, the amount of drug in your bloodstream goes way up and then falls. And when it's way up, you get dyskinesia. When it falls, you're bradykinetic or off. And the idea is that these pumps will create a steady level of levodopa in your bloodstream and dopamine in your brain. And the preliminary results with these both are very promising. And hopefully within the next year to 18 months, FDA will have one or both of these on the market in the United States. It's personally very gratifying to me, a problem I started working on in 1990. And finally, we're starting to see something that's very, very practical for patients. So I think this is a very, very exciting symptomatic treatment. But what about slowing or stopping the progression of the disease? And for this, we look from several sources of information. We look to what we understand about disease biology. We look at what we call translational medicine, which is how we take this biology and find ways to measure it in people. And obviously, we need to test in people in clinical trials and show our results to FDA and the other regulators. A lot of efforts are now focusing on 3 genes, three proteins, synuclein. I'm sure you've heard about these, LRRK2 and glucose-3 residase or GBA. Synuclein is the gene which, if it mutates, you get Parkinson's disease. LRRK2 and GBA are genes which actually are the most common cause of genetic Parkinson's disease, but don't result in people getting Parkinson's disease all the time, which is really fascinating, which is giving us clues as to how to perhaps prevent the disease. So a lot of things can go wrong in the brain and in the cells of the brain, as we know in Parkinson's disease. We know that there's a lot of synuclein in the brain. So we try to, one of the approaches is to try to decrease the amount of synuclein. We also know that synuclein sticks together in clumps, and this form is part of the problem. And so there are drugs out there in testing that are trying to block that process. There are other drugs that are trying to get rid of the synuclein or break it down or degrade it. One company here in Connecticut, here in New Haven, called Arvinus, is a leader in this space. There are also ways of trying just to take the synuclein out of the brain. We're trying to prevent the bad synuclein getting into cells. We're trying to increase the activity of the enzymes that seem to be underactive, or decrease the activity of those that seem to be overactive. And we have a lot of tools in our disposal. We have antibodies. Antibodies are proteins that normally protect your body from germs or other invaders. And so we try to treat these bad proteins in Parkinson's disease as if they were invaders. Vaccines are ways that you're familiar from the COVID story of getting the body to make its own antibodies. We have a variety of molecules that act on genes. And the names of these, the types of molecules, we don't have time to talk about them today, we call them ASOs and siRNAs that can decrease the amount of a protein being made. We have other ways of increasing the amount of proteins that are being made by adding back genes. And this is the company I mentioned here in New Haven that's making what they call degradamers, or molecules that actually help break down bad proteins inside cells. So 8 different ways things can go wrong, and 8 different types of molecules that are in development to try to combat them. But let's talk now about the 2 most recent and unfortunately negative clinical trial readouts. And these are 2 antibodies that were targeting alpha-synuclein. This is a picture of an antibody protein. And an antibody protein has 2 arms, actually 3 arms. This part actually helps the antibody interact with cells in the body and do what it needs to do. And these 2 arms here they attach to the bad thing that the antibody is trying to get rid of. And then the bad thing in this case is synuclein, and it sits here in a little groove right here in the tip of the antibody. And then the antibody takes it away and carries it to specialized cells that gobble it up and destroy it, certain kinds of garbage treating cells in the brain and the rest of the body. So I think I'm going to skip over this because we talked about it, but so this is what we think happens with synuclein and why it causes Parkinson's disease. So synuclein is this little protein, and we know that it has its normal functions in cells, partly to allow cells to talk to each other better. And something happens, we don't know what, that makes synuclein, which normally sits around these little coiled structures, very cute, turn into these little rods, and these rods stick together, and the rods stick together into bigger clumps, and these clumps gunk up the works inside cells. And what we know is that the cells spit them out, and they go on to the next cell, and they get into the next cell, and inside the next cell they cause more clumps, and the clumps go on and they gunk up cells. And when they do that, a whole bunch of different things that you see here on the bottom go wrong, the nature of which we, again, don't have time to talk about, but I think there are at least 6 things that are listed here that go wrong. And the idea of using these antibodies that I just showed you is that they're designed to intercept the synuclein that's going from cell to cell, and therefore to stop the spread of Parkinson's disease around the brain. Now, not all the antibodies are the same. And for example, over here on the right is a picture of the synuclein molecule. And the two antibodies that we're going to talk about today, one called Simpanimab from Biogen, the other prasinezumab from Roche. You can see that they attach to different ends of the molecule. So it's possible, and you read about this in the Alzheimer's disease, well, oh, this antibody didn't work, so these kind of drugs aren't going to work for Alzheimer's disease. But finally, today, somebody announced in the news that their drug actually does seem to work in Alzheimer's disease. And so maybe it's not just as any, maybe the truth is that not all antibodies are the same. And we in the Parkinson field are going to have to be very, very patient. So the New England Journal of Medicine is the big medical journal in the United States, and it's very, very rare for them to publish in the same journal results from two trials when those drug clinical trials were negative. Neither trial showed convincingly that the antibodies slowed the progression of Parkinson's disease. And the New England Journal, if you go to their website, has these very nice one-page summaries that are written at the lay level and I think are very, very instructive. But I'm going to go and show you some of the data that's in these articles. So what these graphs show you is what happens to the UPDRS score. And I think most of you who have been in clinical trials, or even many of your doctors in clinical practice, they'll go through the UPDRS scale. They ask you first questions about how you're doing, about your activities of daily living, and then they wiggle your arms and legs around and ask you to stand up and turn around and flap your fingers and turn your hands over and stomp your feet. And all the while they're grading your responses. And that's the MDS UPDRS scale. And everything you tell them and everything they see you do, they grade on a score from 0 to 4, and then they add everything up. And so you see what happens is that over the first year of the clinical trial, if we look at the blue line, which is the placebo or a control group, people in both of these studies who didn't get the drug got worse by about 10 points. And then if you look at the colored lines, which are the different doses of the antibodies, you see that over a year, it didn't seem to make any difference. And in the case of Simpanimab, these people may actually have been doing a little bit worse for the time being. So this is a very, very, very discouraging result. But again, as I said, this is just a first step, and there are many other antibodies and other drugs that are going to follow. But the Roche team, and here's my friend, Dr. Pagano, who is leading this clinical trial at Roche. This is data he presented at a recent conference. They broke down the UPDRS scale, and they looked, as did Biogen, and Biogen didn't see the same result in their clinical trial. And they just looked at the motor examination, when the doctor's having you flash your hands and turn them over and stomp your feet and get up and walk, that the score for that, and what they found is that in fact, the people who got their antibody seemed to do a little bit better over time than the people who got the placebo. And another thing that both of these groups had in them is they asked the people to wear a wearable device, a smartwatch, an Apple Watch, and carry a certain smartphone and do some tests from time to time, and the watch monitored their movements throughout the day. And as they sifted through all that data, what they thought they saw was that, again, the aggregate data showed that the people who got the drug were progressing more slowly. Now, these results are not what we call conclusive. They're exploratory, but they gave the Roche team enough confidence to go on to initiate a second clinical trial, which will be starting shortly. And I do believe that Dr. Kadimi in Fairfield, who's part of our Yale team, will be participating in that clinical trial. But this shows you what this smartphone and smartwatch test was that they had. They had people do a lot of things on the screen of the smartphone, draw a spiral, tap. They had them turn their hand over back and forth like you do in the UPDRS when you're doing that test. They recorded some speech. They had them hold the phone to record their tremor. And then they looked at what happened when people walked around during the course of the day. And there are some memory tests and other things as well. And that's where all of this data came from. Quite a data analysis feat. And Roche is a pioneer in this space, but this is something that's becoming much, much more common. And all these measurements are very similar. Here you see again the tests in that Roche application. This is another app called Watch PD that's being developed by Biogen and Takeda and the University of Rochester. And here they ask the people to measure their symptoms. They use these little smiley faces to do some maze tracking tasks, again, to do some motor tasks, and record their mood, how they're feeling, a whole bunch of different things. And this is currently being tested in a study that I happen to be involved in. But these apps are not just for research. There are now 3 commercially available FDA-approved apps. And especially if you're a person with response fluctuations and ons and offs, these could be very, very helpful in communicating with your doctor what's going on with you, because you download a version of this onto your smartphone. In this one case, you actually get a watch from the company. This one, Strive PD, I think uses an Apple Watch, and your doctor gets a monitoring dashboard, which you see a picture here and here. And so they can track when you're dyskinetic, when you're off. And then when you go to see your doctor or have a telephone visit, you can better talk very objectively about how to monitor your symptoms. And a similar thing happens here with the Montfort platform. So these are very useful tools that you can use today, and you don't have to be in a clinical trial to use them. Here's just an example. This is one with a research project I was involved with a group in Israel. This very tiny device goes on essentially under a Band-Aid on the small of the back. You don't even know it's there. And it can monitor your movements. This happens to be what happens during sleep. Here's a healthy person, they're moving around, they go to sleep, they turn over several times during the night, they get up out of bed. Here's the Parkinson person, moderately advanced person, moves much more slower, less pronounced movements, getting into bed, turning over much less, much more slowly, and so on and so forth. And we found that this was a very, very interesting index of severity in Parkinson's disease that we may be able to use in clinical trials. People may be able to use this very non-invasively at home. You can wear this thing when you're showering, when you're swimming, when you're doing your kickboxing, when you're getting sweaty riding your bike, anything you want to do. And so this is another very promising avenue for monitoring movement. So what are we doing at Yale? So at Yale, I am part of a project that's going on that I'll tell you about that's very interesting because it involves many, many different laboratories. And we are trying to understand what one of the earliest symptoms of Parkinson's disease tells us about how the disease is developing. And you've probably heard people talk about non-motor symptoms, loss of sense of smell, depression, thinking problems, blood pressure changes, urinary dysfunction, and very commonly sleep disturbance, particularly this thing we call REM sleep disturbance or acting out your dreams. And these things very often start happening before the tremors or the slowness of Parkinson's disease, become apparent. And so in this project, which is involving Yale, Massachusetts General Hospital, the Broad Institute, and Harvard, we're trying to sort all of these things out by understanding especially what happens to people who get REM sleep behavior disorder. Some of you in this group may have experienced this, where you act out your dreams, often very violently, and people can hurt themselves, hurt their bed partners, fall out of bed. About a third of people with Parkinson's disease have this disorder, and it can start many, many, many years before the motor symptoms of Parkinson's. So we ask the question, is there something in the biology of people with REM sleep disorder who, as they have this, but not yet Parkinson's disease, that can tell us about what's going on in the body? And in particular, because we at Yale are very interested in immunology, in what's happening in the immune system and in the microbiome of people with Parkinson's disease. Because it's been shown recently that the immune system becomes activated very early in Parkinson's disease, and that this might be, at least provide a clue as to why Parkinson's disease takes off in some people and not in others. And it's also been shown that the bacteria in the gut may be involved in triggering this whole process. So here's the team. This is a very, very complicated process where we're looking for 90 people. We're about halfway to the finish line. Either people who are healthy controls, people who have REM sleep disturbance, who do or do not have other symptoms suggestive that they're heading towards Parkinson's disease, people with Parkinson's disease and or who don't have REM sleep disturbance. And we're studying their microbiomes, the bacteria in their gut. We're studying their immune system. We're studying their spinal fluid for clues as to what happens that allows Parkinson's disease to develop and evolve. And the theory goes like this, that something microbiome and the bacteria in your gut that does two things, that basically somehow gets the body to react to synuclein, this bad protein in Parkinson's disease, as if it was something foreign. And also it makes it form these clumps that I was talking about, and these clumps travel up the nerves from the gut to the brain. And also by tricking the body into thinking that synuclein is something foreign, it makes the immune system start to react against synuclein. And when you have the synuclein climbing up the nerves to the brain, and the immune system primed to react to the brain, to synuclein, and especially if you have a gene or something like that, or an environmental trigger for Parkinson's disease, when all of this comes together in your brain, that's when you get Parkinson's disease. And so we're trying to understand the interaction of all these very, very many factors. So in order to do this, we need help. We are looking for people to help us out, to donate samples, to donate their time, to help us understand. We're looking for people with REM sleep disturbance who act out their dreams, or people who have been diagnosed with Parkinson's disease in the last 10 years, or spouses, family members, or friends who are willing to participate. We're asking you to complete some questionnaires. Some of these can be done online or over the phone, to give us samples of your stool, swabs of your nose and your gums. Maybe if you think you have REM sleep disturbance, we might want to test you for it in detail, a DAT scan of your brain, and to donate some blood and some spinal fluid through a spinal tap. So if you are interested in this, and I really hope you are, here's Kara. I hope she's listening to the call. She said she was going to be. She's our Parkinson's Clinical Research Project Manager. She's great. She's wonderful. She's easy to talk to. She can answer your questions. And here is her contact information. And we really look forward to meeting some of you in the course of this research. Oh yeah, that's our theme song. For those of you who know and love the Blues Brothers, we need you. There's a lot of other research going on at Yale in Parkinson's disease. Dr. Tanaz is looking at whether cognitive training, things like doing puzzles and things like that, can help with cognitive function in Parkinson's disease. And she's doing this to look at brains. She's looking at brain scans of people before and after cognitive training, and also before and after exercise. She's working with the Rocksteady Boxing Group here in New Haven to do this. And there's another trial, many of you have probably read about ketamine and psychedelic drugs for the treatment of depression. And Dr. Tanaz is also working with a group in the psychiatry department here for people, looking for people who are very depressed in conjunction with their Parkinson's disease, to see if ketamine can help them. So why is it important to participate in research? Well, and for you to get interested on your own, because we can't do this without you. I mentioned bench to bedside and bedside to bench. We really need to understand people with Parkinson's disease, what's happening with their disease, what's happening with their bodies. And we find out that most people with Parkinson's disease really want to participate in clinical research. I think one survey I saw was 90%, but not everybody is getting this information from their doctors. Only about 60% are getting it from their neurologist. And so this is why APDA and organizations like this are so important in spreading the word and helping you learn about ways you can participate in research. And only 20% of people are learning about the research from your doctor. So we really, really need you to reach out, to become interested, seek out sources, and I'll provide some at the end of this talk. Keep checking in with APDA and your support group for information. Again, you are so important with this process. Why? This is a slide from my colleague, Dr. Bloom in the Netherlands. One of the most eye-opening slides I've ever seen in my entire career. He asked people with Parkinson's what's important to them. And he asked doctors what's important about people with Parkinson's disease. And you see how big the differences are. What doctors think is important is not always what patients and their families think are important. So it's very, very important for patients to make themselves heard. And the best way to do this is by participating in research. No one company or institution can do this all. I want to show you a really great example of how drug companies and academic institutions and the FDA are working together. A project I'm very much involved with called the Critical Path for Parkinson's Disease. I'd love to come back and talk about this more, but every pharmaceutical company that's involved in Parkinson's disease research, every major patient advocacy organization, the FDA, the EMA, everybody is involved, sitting at the table, trying to figure out how to do clinical trials better, how to get new drugs to patients faster and more reliably. And I know from where you sit, the prospects seem shaky. We've had our stumbles and falls. The pace of research can seem bradykinetic at times, that maybe researchers are being too rigid in our approaches, but we need, again, I can't say this enough, we need everybody with PD and their families and their friends to help us answer all the important questions. These are some sources, and these will be circulated by Marlene and the team after the call, of where you can find out about information, how to contact us at Yale, the Institute of Neurodegenerative Disorders in New Haven, where a lot of clinical trials are happening, all the major Parkinson research organizations, the Michael J. Fox Foundation, which has an online program called Parkinson Progression Markers Initiative, where you can fill out surveys from the comfort of your own home and be a big contributor to Parkinson's research. All this access is available to you 24/7. So again, back to my friend, Dr. Parkinson, there appears to be sufficient reason for hoping that some remedial process may ere long be discovered, but it's going to happen faster with all of you participating and helping. And thank you very much for your attention and your time. And I hope we have some time for some questions.
Thank you, Dr. Cedarbaum, for a very informative presentation. We
Okay,
have several questions already that have been submitted to us during today's discussion. But if you are participating live today and you have a question for Dr. Cedarbaum, you can ask the question using the Q&A feature on Zoom. Okay, let's go with question number 1
okay. Can
from
patients with-
Judith.
Okay, go ahead, Marlene.
From Judith. Are there comorbidities? Did I say that right? Tights. I said it wrong. Comorbidities. Comorbidities with Parkinson's.
That is a simple question with a very long and complicated answer. So yes, Parkinson, although we like to talk about synuclein very simply, we know that there are other proteins that do build up in the brains of people with Parkinson's disease. In particular, the amyloid protein, the same one that builds up in Alzheimer's disease, is there in maybe a third of people with Parkinson's disease, particularly in people who have thinking and memory problems. There are also interesting immunological comorbidities. For example, people who have inflammatory bowel diseases, Crohn's disease and ulcerative colitis, have a higher incidence of Parkinson's disease than the general population. People who have certain types of cancer have less risk for Parkinson's disease, but people who have melanoma, this is the dark pigmented skin cancer, seem to have a higher incidence, and we don't know if this is related to dopamine metabolism. So the answer there is yes and no.
Okay, next question. Given a genetic variant for Parkinson's in one relative, what can I do to reduce my risk? Are there early signs I should be alert to?
Okay. First of all, if you have a relative with Parkinson's disease and a genetic form of Parkinson's disease, please participate in research, because people like you, only 25% of people who have either the GBA or the LRRK2 gene actually go on to develop Parkinson's disease. So for understanding what happens that prevents somebody with the gene from getting Parkinson's disease, it's almost more important to me for people who have the gene or have the family history and don't have the disease to participate, because we can learn something that can help everybody. So first thing, participate in research, and you can reach out to me. I can tell you places to go. The Parkinson Foundation has a program called PD Generation, where you can get yourself tested for free. You can decide if you want to know the results. If you do, they will provide genetic counseling. If you don't want to know the results, you can be recorded as somebody who's been tested, and your samples, if you give permission, can be used for research. What can you do for yourself to try to protect yourself? Get a lot of exercise. One thing we know for sure. Number two, drinking coffee. Caffeine is known to be protective. Coffee drinkers have lower incidence of Parkinson's disease. Now, I can say that because coffee, by and large, is very safe, unless you happen to have ulcer disease, in which case the acids in coffee can upset your stomach. The other thing, interestingly, that prevents Parkinson's disease or may prevent Parkinson's disease is nicotine or smoking. At this point, I cannot recommend smoking for the obvious health reasons, and clinical trials of nicotine patches and things haven't been successful. So getting a lot of exercise and drinking 1 or 2 cups of coffee a day are probably the best things that we know for helping to prevent Parkinson's disease, if you are somebody who is at risk.
Thank you. We have a question from Das. Will patients who have DBS implants be eligible for synuclein antibody medication when it is approved?
When it is approved, probably yes. During the clinical trial process, there may be in the future some special clinical trials, although right now people with DBS are being excluded from the clinical trials.
Okay. From Rudy and Mary, would being in Tower 1 on 9/11 contribute to getting PD? Rudy was on the 26th floor and the first building was hit on 9/11. The stress and smoke he experienced while running from Tower 1 down on 9/11 could have contributed to his onset of PD? I often wonder.
Well, Rudy, first of all, congratulations on your story of survival. Running down 26 stairs, quite a feat. So let me say that. There's a whole lot we don't know about what the exposures were from in the World Trade Center. So I can't answer that question myself with any degree of certainty. The chemicals that we know that seem to be associated with Parkinson's disease risk tend to be agricultural chemicals and pesticides. And I don't know whether chemicals like that or one similar were released in 9/11 debacle. And I'm not aware of any epidemiological studies that tie Parkinson's disease or any other neurodegenerative diseases to the World Trade Center. It may be a little bit early yet for you to know, for us as a field to know. For example, it wasn't until many years later that we learned, for example, about Agent Orange and neurological problems, particularly ALS. What I would suggest, I suspect you live in and around New York, or if you live in Connecticut, come up to see us in Yale. If you live in and around New York, again, if you contact me through Marlene, I would be happy to recommend people who are might be interested in studying this problem and helping you sort out what might have happened to you.
Thank you, Dr. Cedarbaum. Is there anything else, Dr. Cedarbaum, that you would like to add before we conclude the Q&A session?
Yeah, we've got two other quick questions. One from an anonymous attendee. So, hi, Anne. I'm not aware of links between the Parkinson microbiome and the autism microbiome. We're still at very early days of study. I know there are a lot of genes that are popping up in autism that have to do with general brain development and the way nerve cells connect to one another. And some of these genes are popping up as we study neurodegenerative diseases. I can't think of any immediately offhand, but it's something, again, I would stay tuned to, and I think would be a good topic for a future seminar in the series. And I'm sorry, I'm just not up to date on all of that as I should be. The last thing is there's a question about who to contact at Yale to participate in Parkinson's research, and it's Cara Mead, and I just put her. I just responded to that. I put her information in the Q&A, and I can chat here too, so that you can copy it down quickly. And again, Marlene and the APDA team can make that available. Thank you very much for your interest. There's one more question about the mechanism of coffee as a protective factor. Really, really good question. We think it has to do with the fact that coffee acts somewhat like a neurotransmitter chemical in the brain called adenosine. but it's a very, very interesting story. And again, one that we wish we knew a lot more about. Stay tuned. And probably another good topic for this group: coffee, tobacco, pesticides, what we know about your environment, Parkinson's disease would be a good topic to share learnings with the group and find somebody to talk about that. All really great questions, and shows that I haven't bored everybody all the way through. And so thank you again very, very much for listening. And Kara, if you're there, we have at least one new volunteer.
Thank you, Dr. Cedarbaum. For additional information and answers to your questions and resources, please visit our website. You can also email or call us via the email and phone number on the slide. Okay, for those living in Connecticut, please join us for the APDA Westport Optimism Walk taking place this Saturday, October 1st at Sherwood Island State Park. Pre-event activities and check-in is at 10 a.m. There's still time to register, so please go to our website and learn more about the event and how you can join us. Our symptoms tracker is also available. Contact me and I can let you know how you can get it.
I apologize for not having been aware of that. Great to know.
It's been very popular. On behalf of the entire APDA Connecticut chapter, many thanks to Dr. Jesse Cedarbaum, our participants, and our sponsors, Synovian and AVI, for supporting our program. If you know someone who missed today's program, if you joined late or you would simply like to watch again, you can view a recording on our website at apdaparkinson.org/CT within a day or two. Thank you, everyone, and have a wonderful afternoon.